so I don't forget that. All right, so the first thing I wanted to share with you, and I'll go ahead and share my screen, is this. I just wanted to throw this out there. So in fall, they're going to be offering a course called, it's like a life sciences credit, and it's basically a class. The class number is 90968, and it does fit into the general studies lab requirement, and it is basically a research class. So I know several of you who are going into like biology or medical sciences or anything like that. One of the things that you will find that they ask for is research experience. So at ASU, you're at like an amazing place to get that research experience. You can do undergraduate research through programs such as Enquire or contacting research faculty directly. But another really good opportunity to get research experience is by taking a research-specific class. This is an excellent way to get experience in research methods, just a little more hands-on experience with hypothesis design and everything like that. If you are looking for a lab class that is, I don't want to say it's easy, it's challenging, it's research focused, etc. But it's a really good experience of kind of how grad school works, at least the later years, because you can see from this flyer, there's no tests. So all of your assessment is based on completing your research topics, doing some writing, really good experience for writing, presenting that research, etc. And it only meets once a week on Fridays. So if you are interested in that class, I will post this flyer. Sorry, I don't know what's happening. I do know what's happening. My allergies are going crazy. Anyway, and I ran outside yesterday, so my body's like, why did you exercise? It's a horrible decision, as always. All right, so if you have any questions about this, like I said, I will post this flyer on Facebook, on Canvas. But I do want to mention that the guy that is teaching it, Dr. Sweat, you can contact him at kgsweat@asu.edu. He's awesome. He's hilarious. He's wonderful. And he's really, really good at research. So you'd have a lot of fun. I know you would have a lot of fun being in that class. He's really fun. So, all right, with that, let's go ahead and get started. Again, I will post this on Canvas or send it out in a message if you are interested. I highly encourage it if you're going into any field in biology or medicine or anything like that. This is something you'll want to do. I did a class like this. All right. All right, so jumping into lecture, and I just want to make sure that I have the chat activated. Where did it go? There we go. So if you have any questions at any time, definitely feel free to ask them in the chat, and I will address them. All right, so last week, we were talking about genetics. So the big thing we focused on last week was kind of introducing structure of nucleic acids. So we talked about the difference between DNA and RNA. And we talked about just the fact that DNA has those 4 bases, adenine, guanine, cytosine, and thymine. And then when we talk about those bases, purines and pyrimidines form bonds. So for example, adenine and thymine in DNA, adenine and thymine, or A and T, will bind via hydrogen bonding, 2 hydrogen bonds specifically. And then guanine and cytosine form a bond with each other. And those are 3 hydrogen bonds specifically. And so that's what we call complementary base pairing. It's when those pyrimidines and purines form bonds. Again, in DNA, that's A and T, and C and G that form bonds. In RNA, we don't have a double-stranded molecule. We still have the backbone, which is a sugar phosphate backbone. We still have directionality. So again, we'll revisit that in a second. But we have 4 bases, but the bases vary a little bit from what is present in DNA. So for example, RNA has adenine, guanine, and cytosine, just like DNA, but it does not have thymine. And instead it has uracil. But for all intents and purposes, uracil acts very similar to thymine for RNA in that it binds with adenine. So in DNA, the complementary base pairs are A and T, but in RNA, there is no T. So the complement to A is therefore U. But C and G are still complementary. I don't know where my chat went. What's going on? Hold on. There it is. My chat keeps disappearing. I'm sorry. I don't want it to. There we go. Okay. So that's what we know about DNA. I do want to revisit this just really quickly, that our nucleic acid strands do have directionality. So let me just try this really fast. I'm so sorry. I'm trying to make sure I can see the chat, but for some reason it's popping away. Whatever. Okay. I will check it periodically. Okay, so with our strands, just a reminder that we do have directionality. And for the majority, like I want to say like the directionality that you need to focus on as like left to right, or however way you want to think about it, as sort of like the primary way, where the sugar is up, right? So we see in that figure, we see the O at the top, the oxygen at the top. If the strand is in that direction, that is the 5 to 3' direction. So just a reminder that the 5' direction is referring to the fact that the phosphate, which is kind of exposed, is on the 5th carbon in that sugar molecule. So we call that the 5' carbon, or 5' position. So that phosphate being in the 5' position and sticking up would be the 5' end. And if you just look at the bottom of that, sticking off the bottom, you have the hydroxyl group, or an O and an H, sticking off the 3rd carbon in that sugar molecule, and that indicates that that's 3' end. So shown in the red box on the left, you have a DNA molecule that's 5 to 3, and in the blue box, because this is DNA and it's double-stranded and it's anti-parallel, you have the complement to that 5 to 3' strand is a 3 to 5' strand of DNA. So just a reminder about RNA. RNA is single-stranded, but all RNA is 5 to 3. So that is going to be very important for moving forward, for not only kind of remembering the processes we talk about today, but understanding mRNA. All right, so that was just kind of a summary of nucleic acid structure. And I do want to also remind you of what we're talking about when we talk about transcription and translation. So what are we transcribing? What are we transcribing, basically? And as we mentioned last week, unlike replication, where replication basically takes the entire genome and makes a copy of it all at once. So all of the DNA is copied, which is why we have those little replication bubbles. With transcription, we are not copying the entire genome. So not all of our DNA is being transcribed at once. Instead, we only transcribe little segments of DNA that code for exactly what we need at that time. So those little segments of DNA that code for the things that we need at that time are called genes. And we'll go into this a little bit later. But these genes code for traits mostly. So we would call this coding DNA. So these are going to be things that code for things that are like, the reason it's called coding DNA is because it codes for traits. So it's coding for like eye color or hair color or some sort of phenotypic trait. As opposed to there are segments of DNA that are non-coding. So not to say these are not important, but they just don't code for traits. Typically these are regulatory, and we'll talk about that later. If we're talking about a gene that codes for a trait, there is a beginning and an end to a gene. So a little segment of DNA, there's a beginning and an end of that segment. The beginning of that segment of DNA, the beginning of that gene is called the promoter or the promoter region. So promoter, it's promoting transcription. And the end of that gene, the end of that segment of DNA is called the terminal region. So just think of like, or the terminus, the terminator is also used in some textbooks, even though I think that's hilarious. But you can think of the promoter, it's promoting transcription, where the terminal region is the terminus. Think of that as the end. All right. So I hope everybody is able to hear me right now. I just got a little message about my internet connection not being super stable. So if you have any questions, let me know, right? I'm trying to make sure that everybody can hear me. But if you have questions or if you're having trouble hearing me, write it in the chat so that I can see. All right. Again, this will be recorded, and if the connection gets really bad, what I'll do is I'll record it later and post that as well. Okay, so we feel good about the structure of a gene. Now, let's go through how we take that gene and we transcribe it into mRNA through the process of transcription, and then how we then take that transcript, which is in the form of mRNA, and turn that into, we're going to be talking primarily about protein synthesis. So I do want to mention, you can see on this slide, that, goodness, I'm so sorry. I have something stuck in my throat and just wouldn't let go away. Okay, all right, so with gene expression, again, the first thing we have to do is take DNA and turn it into RNA. And you'll notice that this is divided into 3 steps: initiation, elongation, and termination. So basically what that means is that we have to start this process, and then we have to elongate a molecule, and then we have to stop the process. So these are all active things that happen. And I want to kind of key you in here to the terminology. With transcription, what we are doing is we are taking DNA and turning and making a copy of that code, or basically taking complementary copy of it in the form of RNA. So whenever we do that, we have a template, right? Think about it as like you see something that's in a textbook and you're copying it onto notes. Your notes would be basically the mRNA. So you're making a copy of what's in the textbook. And here, when we're talking about elongation, what we mean is we're elongating a molecule. So in this case, with transcription, because we're taking DNA and turning it into RNA, or basically making a copy in the form of RNA, what we are elongating is RNA, right? So with transcription, you have initiation, elongation, so making that RNA molecule, and then we end, termination. And then with translation, what we do is we take that RNA copy and we turn that into protein. So we'll talk a little bit more about that later, but again, there's same 3 steps, which can be a little confusing: initiation, elongation, and termination. In this case, we are starting, but what we're elongating is, in this case, a strand of amino acids linked by peptide bonds. So just a reminder that proteins are made out of amino acids, and those amino acids basically link together and they form all those complex interactions to fold. So with transcription, we're elongating a strand of RNA. But with translation, we're elongating a strand of amino acids to make a protein. So similar terminology with steps, but you need to know what happens during each of those steps of both of those processes. All right. Okay, so moving on, let's start talking about what happens during transcription. All right. Okay. So just a reminder that what we're doing with transcription is we are taking DNA and we are going to synthesize or create a complementary strand of mRNA. So what we're going to do is not copy the entire strand of DNA, but all we want to do is make a copy of RNA based on a segment of DNA that is a gene. So what you're only going to copy is a gene, which remember is from a start to a finish, promoter to terminal region. All right. So we get into the first step, which is initiation. So how do we start this process? So the questions here are basically like, we know we have to have an enzyme to do this. It has to copy it. But how does that enzyme know where to bind, right? So how does our enzyme know where to go? And the answer is that there are features of genes, that promoter region, that allow those enzymes to know where to bind. And again, it's features in that promoter region. There's a lot of other complexities, but we're not going to go into that. But basically, the promoter region, the start of that gene, has features that basically tell or say that that's where our enzyme needs to bind. So our enzyme is RNA polymerase. So if you think back to what we talked about when we talked about the last lecture, reproduction and inheritance, when we talked about DNA replication, we talked about taking a strand of DNA and making DNA, synthesizing DNA. And in order to do that, we used an enzyme called a polymerase. So just a reminder that a polymerase is an enzyme, ASE, so polymerase, can go up here. ASE, remember that tells us it's an enzyme. And remember that a polymer is a large molecule. So this tells us that it's an enzyme that builds large molecules. And it always tells us what the large molecule it's building is, not what the template is, what it's creating, right? So in this case, we use an enzyme that creates large molecules. And in this case, the large molecule we're creating is RNA. So the reason I redirected you to reminded you of replication is because during replication, we used DNA polymerase because we were taking DNA and making a copy in the form of DNA, a complementary strand of DNA. So DNA polymerase makes DNA, RNA polymerase makes RNA. And since with transcription, we're taking DNA and making RNA, we need an RNA polymerase. So RNA polymerase is our enzyme here. And what it does, and you can see here on the top, we have a strand of DNA. And you can see shown in yellow is the promoter region. The end of that gene is the terminator, shown in orange. And what happens is our enzyme, RNA polymerase, binds to the promoter of this gene. So it binds to the start site of this gene. And you can see here in the box over here, it says that when it binds to the promoter, it melts a short stretch of DNA. I hate the terminology melts because that's a little weird, but basically what they're getting at is that it denatures or separates or opens that strand of DNA. So that's important, again, because if we want to make a copy of one of these strands, we need to open up the strands so we can use that strand as a template to make a complementary strand, in this case in the form of RNA. So it is really important when you're thinking about these processes to understand what enzyme is responsible for opening up the strands of DNA. So if you remember with replication, so I'll write this down as just a reminder, with replication, it was DNA helicase that opened, what's going on? That opened up our DNA strands, or opened up or separated our DNA strands. Whereas during transcription, it's actually our enzyme that does the copying, which is RNA polymerase, that opens up or separates those two DNA strands, basically making the template exposed. Right, so that's really important. I know that you have a question about that on your Unit 2 exam. I also know that you will be asked to compare these processes, replication and transcription, on future assessments. So it would be very prudent to know, compare in transcription versus replication, which enzyme functions to separate those strands. Again, in replication, DNA helicase, and in transcription, it's RNA polymerase. RNA polymerase practically does everything in transcription. Okay, feel good? I think so. If you have questions, pop them in the chat and we can answer it. Okay, so in order to start, basically what we did is we bound RNA polymerase to the promoter. So we're ready to go. And we'll look at what happens next. You'll notice that in this figure it shows sigma. We're not going to discuss sigma. If you need to learn that, you'll learn it later. All right, so we have bound our enzyme to our DNA. We're ready to go. So now what we need to do is, again, take one strand of DNA, and what we want to do is make a complementary copy, a strand of RNA. So in order to do this, in order to make a strand of nucleic acids, we basically have to have nucleotides, so those A, T, C's, and G's. Well, in this case, we're building RNA, so it would be A, U, C, and G that we would use, and then just bind them together to create a strand of RNA. So in order to know the sequence of nucleotides we need to have a template, and that's the purpose of DNA here. So if you can see from this slide, this is very, very, very important here, is that what is happening is the two strands of DNA, right, they're like this, are being separated by the enzyme. So if you look behind that green enzyme, you can see that just a small segment of that DNA is being separated. And you can see that what the enzyme is using, it's a little bit difficult to see, the enzyme is using the 3 to 5 prime strand of DNA, right? So RNA polymerase is using the 3 to 5 prime strand of DNA as a template in order to create, remember, complementary means you're going to have an anti-parallel strand. So if you're using a strand of DNA that's 3 to 5 to build a strand of RNA, your RNA is going to be in the correct orientation, which is 5 to 3 prime strand of mRNA. Okay, so again, it's a little bit hard to see in this figure, but you can see the blue line is your 3 to 5 prime strand of DNA. RNA polymerase is opening it up, exposing that little template. Those little black lines are your nucleotides, and you can see that this sort of like, I guess, pink strand is going to be your mRNA. If you're using the 3 to 5 prime strand, what you're building is going to be a 5 to 3 prime strand of mRNA. That is so, so, so important. We'll talk about why later. Okay, so basically what happens during elongation is, again, you initiated this process by binding your RNA polymerase to the promoter. That opens up the strands of DNA just a little bit, and then what can happen is that it can travel along your DNA, elongating or creating a strand of mRNA as it goes. So again, it uses the 3 to 5 prime strand of DNA to create a 5 to 3 prime strand of mRNA. Messenger mRNA specifically, but the same process we're looking at here would be used to create any type of RNA we need. But right here we're talking about protein synthesis, so we're talking about mRNA. Okay, so that's elongation. What we do is we keep going down the strand until we meet the terminal region, or the terminator, and basically it's pretty anticlimactic here. It just goes and goes and goes and goes and goes. Once that RNA polymerase reaches the terminal region, that little orange box right here, it just pops off and then disassembles. So it is a little bit more organized than that, but we're not going to go into it. So you can see RNA polymerase pops off. When that disassembles, what we're left with is a 5 to 3 prime strand of mRNA. We create an mRNA. Yes. All right. So what we did, just to revisit, initiation of transcription, RNA polymerase binds to our promoter, opens up the DNA just a little bit. Then we use the 3 to 5 prime strand of DNA as a template to create a 5 to 3 prime strand of mRNA, just using complementary base pairs to build that. And then we reach the terminal region, disassemble, and we have our mRNA. Okay. So now it's important to note that within a gene, we have segments of DNA that basically contribute directly to the protein. We also have segments of DNA, or of that mRNA, that are not really necessary. They're kind of just, I don't want to say spacers, because they do play an important role, but in this process, they serve as sort of basically like little segments of DNA where mutations can happen and it's not a huge deal. So these are called introns and exons. So these, again, I don't want to kind of throw away the purpose of introns, but what I will say is that introns are non-coding, which means that they are segments of DNA and thus mRNA that are not going to contribute directly to a phenotype. So they have very important other functions, but just not in the process of turning mRNA into protein, at least directly, coding-wise. So what I want to show here is when we finished transcription, we do have a strand of mRNA, but that mRNA has to be processed before it can enter translation. So this is what we're talking about here. We're talking about mRNA processing, very exciting. So I'm kind of blowing it up there. And there's two things that are really important that happen during mRNA processing. First, we splice. I don't know why I have that listed second, but we splice. And then the other thing that we do is we add a cap and a tail. So a cap and a tail, let's talk about that first. You can see in this picture, so just kind of orienting you to this picture. In blue, we have our DNA. So again, DNA can be organized into coding and non-coding regions, those introns and exons. Both intronic and exonic regions are transcribed. So whenever we get our mRNA, which is shown in pink, we do have mRNA that has exons and introns. And you can see one of the first things that happens after transcription is we add these little yellow parts on the beginning and end, the 5' end and the 3' end of that mRNA transcript. On the 5' end, we add what's called a cap. And this is just a series of G's, nucleotides kind of added to the end. And then a tail, which is on the 3' end, is a really, really long sequence of adenines. So on the 5' end, you just have a series of like guanines. And on the 3' end, you just have a really long tail of adenines. So what's the purpose of these? There is a lot of different purposes, and I will say that we don't 100% understand all of the purposes, but what I can tell you is two really important things. Basically, they serve as protection. So if you look at your mRNA, everything that is in pink is, or at least dark pink, is going to be important for being turned into a protein. So that mRNA that's in pink is really, really important to protect because it's going to contribute to creating your enzymes or your proteins. The mistakes in that process would be genetic mutation, which can potentially be harmful. So we need to protect our RNA. As we'll talk about a little bit later, well, you can kind of see here, transcription occurs where the DNA is. DNA is in the nucleus. So the nucleus is protected by a membrane. It's a very kind of separated region. So transcription occurring in the nucleus means that that mRNA is created in an environment that's very safe from a lot of mutations or harm. But what happens is that that mRNA has to, in order to be translated, it has to associate with a ribosome. And ribosomes are in the cytoplasm, or for free ribosomes, or they might be attached to the rough endoplasmic reticulum for bound ribosomes. Both of those, any of your ribosomes, except for mitochondrial ribosomes, all of your ribosomes are outside of the nucleus. So this means that in order for an mRNA transcript to be translated, it has to leave that protective area of the nucleus. And you can see that happening on the bottom of this picture. So the issue with this is that it kind of opens the door for damage. So one of the purposes of adding a cap and a tail is basically to protect the coding portion of the mRNA from any damage as it exits the nucleus. And also, another thing that it does is, one of the questions I get a lot is, okay, like mRNA leaves the nucleus and then associates with the ribosome, like how does it know where to go? And the answer is in those regions, I want to say it's in the tail region, but in one of those regions, there's something, they literally call it like a zip code. And what it does is it helps that mRNA know where to go. Does that mRNA need to go to a free ribosome so that it stays in the cytoplasm? Or does that RNA need to go to a bound ribosome so that it can be produced in a vesicle and therefore become part of the cell membrane or become part of another organelle or something like that. So the cap and the tail are protective, but also direct that mRNA basically like where to go, right? Which ribosomes to associate with. It is important to note that the 5' cap and the 3' tail are not translated. So they do just kind of exist. I also like to point out another really important thing about the cap and the tail is that kind of applicable right now. A lot of viruses actually are in the form of mRNA. So that actually does include coronavirus. Coronavirus is an RNA virus. So once it gets into your cells, it looks remarkably like mRNA. So it's really, really easy for that virus to then associate with a ribosome and then translate all its proteins and make more virus. However, one way our cells can recognize viruses as not belonging, right? Our immune system is like, this doesn't belong here, is that viruses do not have, not all viruses. Most viruses do not have a cap or a tail. So one way your body can say, is this my mRNA that needs to be turned into a protein, or is this like viral RNA, which is bad, is to say, is there a cap and a tail? Right. So again, that's just another purpose of the cap and the tail, or at least that's like an additional benefit to the cap and the tail, I should say. All right, so again, we translate our DNA into RNA, and one of the first things we do is we add a cap and the tail. A 5' cap, which is a bunch of guanines, and a 3' tail, which is a bunch of adenines. And again, this is present primarily on eukaryotic mRNA. All right, so after that, the next thing we do is we want to make sure that our mRNA strand only includes nucleotides that are going to contribute directly to the creation of protein. So we don't need those introns in it anymore. So we do what's called splicing. So splicing is a combination of both cutting out and pasting together. Cutting out those non-coding regions or introns. So you can see that's what's happening in this figure right here. You have those light pink areas, those are your non-coding areas being kind of spliced out. And then what happens is you have the coding regions, the exons, kind of pasted back together. And you can see it says the exons are spliced together. So technically, even though it seems a little bit counterintuitive, splicing is subsequent to intron removal. So what happens after you add the cap and the tail, you remove introns, and then splicing is actually connecting the exons back together. So what happens is you go from mRNA post-transcriptionally, you go from mRNA that contains introns and exons, add a cap and a tail, you remove those introns, and then you splice the exons together. And what you're left with is a strand of mRNA that has been processed so that it has a cap and a tail, so it's protected, and also has a zip code so it knows where to go, and also only contains nucleotides that will contribute to a phenotype. All right, so that's where we are. This is just another figure kind of showing that splicing taking place. So you can see your DNA is shown in blue. You have coding and non-coding regions. Again, the exons are the coding regions. The introns are the non-coding regions. Via transcription, you make a copy of this in the form of RNA. You still have introns and exons. And so via processing, you add a cap and a tail, which is not shown here, but also via processing, what you're doing is removing those introns. So then for translation, you only have mRNA with coding portions of the strand. All right. This is literally just another figure of that. Just all about lots of figures showing the exact same thing. Okay, so I actually have, I think, hopefully, I figured out sort of the polling feature. So why don't we try a clicker question? What do you say? Okay, so here is your question. The question is, what is the promoter region? And you have 4 options, A, B, C, and D, and I'll give you a minute to answer. All right, ready, set, go. Good job for everybody going, keep going. What is the promoter? Make sure you read the answers very closely. Right. Think about where the promoter is. You can change your answer, I'm pretty sure. All right, so we're going to stop there for this one. Everybody feel good? Okay, we're going to stop. Thank you for answering. Good job. All right, so end this poll. Your polling box disappeared. I'll see if I can do it again. Don't worry about it. Okay, so share results. Okay, so these are our results, and it was a really good tie between A and D, which everybody got, right? That it's a place where RNA polymerase binds and initiates transcription. But I see the big difference here, and the most important difference here, is that the answer is actually D, because remember that the promoter is in the DNA. So A says it's in the RNA, but promoters are in the template DNA strand. So the answer is D. All right, and there you go. D, you couldn't find, that's okay. We'll do it again. We'll have a lot more. It's just to kind of, not a big deal. Okay, cool. All right. We'll keep trying that, so don't worry if you couldn't find it. We'll try it again. Okay. So, feel good? Any questions about the promoter region? Does that make sense that it's in the DNA strand and that's where RNA polymerase binds? So just to kind of show you that. You have your DNA strand of a gene and you have your promoter region. All right. So let's go ahead and move on. We will try that polling thing again. So don't worry if you couldn't get it to work. We're going to try it again. We'll keep trying it. Not a huge deal. Okay. So let's move on. We talked about transcription. Here is a summary of that entire process. So during initiation, you have RNA polymerase. We're not talking about sigma factors. Don't worry too much about that. But RNA polymerase binds to the promoter. DNA is denatured, or you can say strands are separated. It's a little bit more kind of easy way to think about that. And then sigma factor releases. Again, we didn't really talk about sigma factor, so don't worry too much about that. I think I have high hopes to talk about it someday, but we just don't have time. All right, so RNA polymerase binds to the promoter. I didn't mean to do that. Binds to the promoter, separates those strands of DNA, and then what happens is RNA polymerase reads a 3 to 5 prime strand of DNA into a 5 to 3 prime strand of SS, is single-stranded complementary RNA, which is your mRNA. All right, and this continues until it reaches the terminal region. Once it reaches the terminal region, RNA polymerase detaches, and then your mRNA is processed. So that means the cap plus the tail plus splicing. And then importantly then, what happens is our mRNA then leaves the nucleus. Right. So obviously, this is important. It leaves the nucleus in eukaryotic cells because eukaryotic cells have a nucleus. Prokaryotic cells don't have a nucleus. So there's a little more kind of cool things that happen in prokaryotic cells. But in eukaryotic cells where you have a nucleus, a very important thing that ends transcription is that mRNA leaving the nucleus and entering the cytoplasm where it'll associate with a ribosome. All right, so we understand transcription. Are there any questions about transcription? Or we feel okay? We're feeling all right. I'm not seeing any questions. If you do have questions, just pop them up, no big. All right, so let's go ahead and move on to the next step, which is translation. All right, so with translation, what we're doing is we're taking that mRNA we just created via transcription, and we want to turn it into protein. So before we do that, it's important to understand a couple of things. So first off, a little bit about the structure of mRNA and how it's used to create amino acids. So mRNA is divided into basically three nucleotide sequences. So you can see if we have an mRNA strand shown in orange here, you have a sequence of 3 nucleotides, say A, U, and G, adenine, uracil, guanine. Again, this would be 3 to 5 prime direction. 3 nucleotides. Every series of 3 nucleotides is called a codon. All right, so if we have this mRNA strand, this would be a codon. The next 3 nucleotides would be a codon. The next 3 nucleotides would be a codon. The next 3 nucleotides would be a codon. So as opposed to transcription, where if we have a strand of DNA, you go nucleotide to nucleotide for complementary base pairing. Whenever we do translation, we're not going nucleotide to nucleotide. Instead, we're going a series of 3 nucleotides codes for an amino acid. And remember that a protein is basically a long strand of amino acids. So it's codons, which are 3 nucleotides that code for amino acids. A couple of things that we need for translation. We need a molecule called tRNA. This is also called transfer RNA. We will look at this in a minute, but basically what that does is it brings the amino acids to the ribosome mRNA complex, which will eventually contribute to the growing amino acid strand, which will be our protein. So that is the function of tRNA, which is transfer RNA, is it's basically going to bring the amino acids to, basically you have a codon, again, whoa, look at this, you have a codon, like 3 nucleotides, A, U, G. That codon codes for an amino acid. So how we get that amino acid to that strand is we have to have what's called a transfer RNA. So we'll look at this here in a minute, more specifically. We also need ribosomes. So we have talked about ribosomes in the past when we talked about cell biology. We talked about the fact that we have free ribosomes and bound ribosomes. Those are just location-based, although it is important to distinguish them because it's important to distinguish what type of proteins they will make. So reminder, so let's just type it out so we know, so we remember. Free ribosomes are in the cytoplasm and they create proteins that will stay in the cytoplasm of the cell, whereas bound ribosomes are attached to the rough ER, or what makes the rough ER rough, and they create proteins that will be wrapped in a vesicle, which, remember, vesicles are made out of membrane, meaning that they can associate with membranes. So they can pass through membranes or incorporate into membranes. So they're going to make proteins that leave the cell, proteins that incorporate into the plasma membrane or into membranes of organelles, or proteins that enter organelles. Just basically, like, anytime a protein is going to function, sort of have to interact with a membrane some way, either to incorporate into it or pass through it, it has to be wrapped in membrane. So that would have to be created at a bound ribosome. So those are our required components for translation. Obviously, we need our mRNA as well, so we should probably list that, right? mRNA, which is our, which basically serves as, you know, like our transcript for this process, but I'm not going to write transcript because I don't want to confuse anybody. All right, so our required for components for this process are obviously our mRNA, which serves as our template for this process, our tRNA, which serves to bring amino acids to the ribosome, and then ribosomes, obviously, which we have 2 types. Okay, any questions right now? So these are just all the components that we need for translation. Feel okay? All right, I'm not seeing any questions, but if you have questions anytime, just pop them in the chat. Okay, so those are what we need. So before we get into it, I want to talk a little bit about the structure of tRNA. So remember that this is telling you that it is composed of RNA. So this is a single-stranded RNA molecule. It has it's created out of the nucleotides A, U, C, and G. But you'll notice that it looks double-stranded. It's actually still RNA, it's still single-stranded, but what happens with tRNA is it actually gets folded and it interacts with itself. So it's still single-stranded. It's not composed of 2 anti-parallel strands, but what happens is that single-stranded molecule starts folding in on each other. And it folds in on itself in a really cool kind of pattern. And what you can see is what happens is it folds in on itself in a way where it's literally called tRNA because what it does is it transfers amino acids to the ribosome. So it's called transfer RNA. But I also think it is so cool that it's called tRNA because if you kind of look at its crystal structure, which is a sort of a cartoon of that structure, it literally looks like a T, right? So kind of cool, a little T, tRNA. You can see that the structure of this is extremely important because on the tip of this tRNA molecule, you have an amino acid attached. So basically tRNA carries amino acids on the top of the T, and then on the base of the tRNA molecule down here, you can see that every tRNA actually has a sequence of 3 nucleotides, and this is called an anticodon. So it's important to note that an anticodon is a 3 nucleotide sequence that is complementary to the codons specified in the mRNA. So let me write this out so that it's a little more clear. All right, so tRNA has an anticodon that is complementary to a codon that is present in the mRNA. Okay. So what you're looking at here to orient yourself to this slide is you'll see in the mRNA strand, this picture is showing a codon. So I'm just going to specify that this is an example, right? So in this picture, you see that the mRNA has a codon of CCG. So we need to know which amino acid that codon is specifying. And really the only way we can sort of like read stuff genetically is via nucleotides. So in order for a tRNA to know which amino acid it needs to bring, it's going to have to be able to read that genetic code. So therefore, if mRNA has a codon that's CCG, we know that the complement to C is G. Oops, anti-codon. My husband is Cody, anti-Cody. If our mRNA has a codon that's CCG, the complement to C is G, the complement to C is G, and the complement to G is C. So if our codon is CCG, the anticodon, or the tRNA that would recognize that codon, would be GGC, right? It's all complementary. So basically what happens though, we'll look at this, is that the tRNA that carries the anticodon, GGC, codes for the amino acid proline. Let's get this a little bit more clear. So tRNA are specified based on what amino acid they carry. So for example, this tRNA carries proline. But how it carries it and how it matches it is based on that anticodon-codon complementary base pairing. So we'll talk about this here in a minute. This can be somewhat redundant, and in some cases this is referred to as being degenerate for some reason. But this can be somewhat redundant. So for example, there are several tRNAs, not only the tRNA with the anticodon GGC, but there are several tRNAs that carry proline. So this can be somewhat redundant. But it is important to note that if mRNA has the codon CCG, that brings, because of complementary base pairing, if you have a codon that's CCG, that will bring or attract a tRNA that has proline. So those codons specify what amino acid needs to be brought to the ribosome, because the codons tell what tRNA to come associate with that. So we'll look at this process take place more, but that's sort of like the technicalities of how tRNA works. So speaking of codons, like I mentioned, there is some redundancy. This is wild every time I turn it, bring it up, but I do want you to be able to read. Technically, this is sort of what people talk about when they talk about the genetic code. This is called the codon table, and it basically lists all of the possible 3 nucleotide sequences that can make up codons. So for example, it is important to note that these are codons listed in this table. So what that means is that these are the 3 nucleotide sequences that are present in mRNA. So I really, really want you to note that, because a lot of students, when they talk about codons and anticodons, will be a little confused over what this table is talking about. Is it talking about codons or is it talking about codons? This table is talking about the sequences present in mRNA. It's talking about the codons. So what I really want you to note here is that as far as knowing what an anticodon is, all you need to know is that the anticodon is on tRNA, and it's basically how we have that we know which tRNA to bring to the mRNA. There's really no reason to memorize anticodons or anything like that. You don't need to memorize this table either, right? So don't worry about memorizing it. But it is important to note that what this table is saying is that the codon, right? So if we look at this, the codon is in this mRNA strand. If you have a codon in your mRNA, say your codon is UUU, this will attract a tRNA carrying the amino acid phenylalanine. Again, you don't need to memorize that, but I do want you to know that what this table is specifying is codons, which are in mRNA. And this is just telling you which, based on what codon is in mRNA, that's what amino acid will be brought. So the way the codon table is organized, and it's a little bit easy to read, is that you can organize it based on the first letter of the codon sequence. So if your first letter is a C, you'll look in this row, and then along your columns are organized based on the second letter present in the codon, and then finally you can organize it by the third letter. So for example, sorry, I see your question, Erin. That's a really good question. Let me get to it in a second. But for example, in the table, if say you have a codon that's like A, U, G, the first letter is A, second letter U, third letter G, so we know AUG is right here. And this tells us that if the codon present in the mRNA is AUG, this is going to attract the amino acid called methionine. All right. So I had a question in the chat. The chat says, the reading talks about a clover structure. Is that significant to the tRNA function? Yes. I guess when you look at the tRNA structure, it's not really easy to see here, but let's go ahead. Ooh, I have a lot of windows open. Clover. Well, if you look at tRNA structure, right? Hopefully everybody can see this. That's showing it upside down for some reason. I don't know why it's upside down in this picture. But basically what you can see here is that the clover structure is very similar to like the T structure. So in the case when I was talking about tRNA being in a T structure, I'm thinking of like a lowercase t. You can see that here where it's the same thing, it's just kind of talking about it a little bit different. It's kind of a lowercase t or a clover-like structure. That's talking about the same thing. So yes, I guess to answer your question, that is significant. The clover-like structure is significant because it's on sort of the base of that T or clover molecule where you have your anticodons. You can see that in the picture. Let's see if it's a little bit better. Yeah, so you can have the anticodon, which is on kind of the base of that clover-like structure. And then at the top of that, where I said sort of the top of the T, but in this case, like the tip of the clover or whatever, is where you have your amino acid carried. So it's basically the same thing. Yeah, good question. Yeah. So yes, I will write that down. Structure is always very important to function. Good, good, good question. I guess I'm going to go ahead and write here. Or you can also see, again, that the clover-like structure, that's the same thing as like the T-like structure. Good question. All right, so again, we have our codon table here. And again, I just want to iterate a million times the fact that this table is telling you, talking about what amino acids are encoded by the codon in the mRNA. So for all intents and purposes, forget about the sequence of the anticodon. All right, anticodons are complementary to codons. It's just how what tRNA knows where to bind, basically. All right. So this is kind of just showing how this process takes place. Again, we have our mRNA shown at the bottom. mRNA is separated into 3 nucleotide segments called codons. So here we would have a codon that's AUG. Based on that, the tRNA carrying the anticodon that is complementary to that, which is UAC, would come and bind, right? So that tRNA carrying methionine, right? So again, so first if we have AUG, we look at that at the table, AUG codes for methionine. So that's how we know if we have a codon that's AUG, it's the methionine carrying tRNA that's going to bind, is just because of the fact that we have that complementary base pairing. All right. We can go next if we have the codon that's CGA. We look at our table, CGA codes for arginine. That's how we know that the tRNA carrying arginine is going to bind to this codon, and so on and so forth. Okay, so that's kind of how we recruit those tRNAs. So a little bit more about the codon table. You'll notice that I have a couple of these boxed in green or red. So something that's very important to note is that just like with transcription, we have to know when to start translation and when to stop it. So where is the beginning of the mRNA molecule and where is the end? And we don't have a promoter or a terminal region in mRNA because our mRNA is separated into codons. So instead, what we have in mRNA is basically a start codon, and then we have what are called stop codons. So where do we start? Where do we stop? We start translation at a start codon, and we stop translation at a stop codon. Our start codon is always going to be AUG. AUG codes for methionine, which is going to be our start codon. This doesn't mean that there's only going to be one AUG or one methionine in any protein, right? It's just that the first AUG present from 5 to 3 in an mRNA strand will be where translation starts, and I'll reiterate that later. Translation will then proceed all the way down the codons until it reaches one of 3 stop codons. So we have 3 possible stop codons. We could have UAA, UAG, or UGA. So those are just 3 possibilities. They all code for where translation will stop. So you can see that here. Again, I have AUG listed as the first codon. It codes for methionine. That's where translation starts. We would read codon to codon to codon all the way down the mRNA strand until we were to reach one of these stop codons. All right. So we have a couple of minutes left. What I want to do really quickly is try the poll again. So this says anticodon is the term applied to what? So this is quiz number 4. I'm going to go ahead and launch. Hopefully everybody can see that. Go ahead and answer what you think the correct answer is. And I'll give you a minute to do that. Hopefully everybody can see the quiz. So anticodon is the term applied to what? Okay. So we've got a minute. So anticodon. So I'm going to go ahead and stop it. I see some of you changing your answers. That's fine. If you're moving around, I am going to go ahead and stop it so I can share the answers. Okay. All right, so the answer is C. Good job. All right, so an anticodon is the term applied to, that's correct, the part of the tRNA that interacts with a codon. So again, we look at that here. It's just where we have a tRNA. The anticodon is directly where the tRNA interacts with the codon. It's basically the anticodon is how the tRNA knows where basically the sequence of amino acids to bring to our mRNA strand. So good job. All right. So this is, I think, we only have about 4 more minutes. So I think what we're going to do is I'm going to stop there before we get into the process of translation, because I want to leave the last like 4 or 5 minutes for any questions. So questions related to the lecture, questions related to what's going on, what's coming up, anything like that. And then the inheritance problem set. So I had a question, great, let's look at it. So question on the problem set, do you round up the test statistic to 2 decimal places and the p-value to 3 decimal places? Let's see, I forgot. Yeah, you can do that. I mean, I would have your test statistic at around 2 decimal places and the p-value to either point, kind of 0.0, yeah, 2 or 3 decimal places. That would be fine. I'm not really grading you based on significant figures or anything like that, so don't worry too much about it. Yeah. I hope that answers your question. Good question. Yeah, we didn't get to talk about significant figures. I'll leave that for the chemistry folks. All right, good question. Any other questions about anything? Anything you'd like to talk about? Anything like that? I'm happy to do it. I will also be online today from 11:30 to 12:30 for office hours. Those are open office hours, so a lot more people might be there. Sometimes another professor pops in, but if you also want to have private office hours, I can arrange that. I can also answer questions via email. Just a note, and kind of like I've been sending out, I've been fortunate in that I've been able to arrange childcare on like Tuesdays and Thursdays during lecture time and during office hours. But after 5, I don't have that, so my replies to emails and everything like that might be a little bit delayed, and I don't have the ability to pop on Zoom after 5. So if you have questions, if possible, try to get them to me earlier in the day, because that gives you just more time to answer them. And another question also. For the proctored exam, are you still allowed to use your notes or to have the PowerPoint up? I am not sure. No one was able to answer me about whether you can have the PowerPoint up, but what I would recommend for the proctored portion of the exam, okay, thank you, Noelle. So you can't have the PowerPoint up. What I would recommend doing anyway, though, because scrolling through the PowerPoint will probably isn't the best way to get to the information anyway, what I would recommend doing is going through your terms and conceptual questions and answering those on paper and using that as your notes. So that's what I would recommend. You can, if you want to, pull up the PowerPoint on like your phone, if you're taking the quiz on the computer, or if you're taking the quiz on your phone, you can pull up the PowerPoint on your, oh, you can't take the quiz on your phone because you need Respondus Lockdown. But if you have Respondus Lockdown open on your laptop or your computer, you can pull the PowerPoint up on your phone if you want to. But again, it's timed. You don't have a lot of time per question. So what I would recommend is just having a really good set of notes, maybe taking the terms and the PowerPoint and having that written out, maybe having the problem set kind of written out to where you understand how to do a Punnett square and stuff like that is what I would recommend. Yeah, thank you, Noel. Right. I hope that answers your question. Right. For part 2 of the exam, is there a specific format that the phenotypic ratio should be written in? If there is a 0 chance of a phenotype being present, do you include that 0 in the ratio? Yes. I guess, yes. Typically, the way you want to write your phenotypic ratios is you want to write dominant to dominant homozygous to heterozygous to recessive homozygous, which would basically be like PP to PP to PP for monohybrid. For dihybrid, it's the same. It's sort of like your dominant phenotypes. So it would be like, let's see, how do I write this out? Both traits dominant to heterozygotes to hetero to both traits recessive, if that makes sense. So as for the dihybrid cross, I guess a way, okay, good, yeah, good. For the dihybrid cross, the order of hets that you include in the phenotypic ratio isn't really important. So if you remember, like if a trait is inherited in a Mendelian fashion, the phenotypic ratio you should expect would be 9 gametes that have both dominant traits expressed, 3 to 1, the 3s being where like one of the traits has the dominant trait expressed and the recessive, and then the other is like recessive or dominant, but it doesn't matter what order you put those in. But you do want to have it usually dominant, het, recessive. Good question. All right. And yes, include a 0 in that if there is a 0. Any other questions? Go ahead and ask. Talmud, good. Okay, so when it asks to give the phenotypic ratio, can you write the phenotype and number? Yeah, totally. That's fine. Honestly, whatever makes sense to you. As long as you have, like, so sometimes what I will do is I will write, like, just to specify, I'll say, like, so how would I do that? So let's say purple to white, and then I would say, like, the ratio of purple to white would be like 3 to 1. So I would just write purple to white so that people know the numbers that I'm talking about. So you can do it in parentheses like that, totally fine, or you can do it and just like specify with words what your ratio will be and then have the numeric ratio after that. However you want to do it, as long as I understand it, that's like the most important thing. So just to be straightforward with you, I will be having a TA help me grade these. So it is really important to be very specific with your answers so that, I guess I can have this. It is really important to have your answers very specific, just so the TA, me and the TA understands what